Hello everyone. Today, we are pleased to present a paper titled Non-Histone Lactylation, the New Frontier in Cerebral Ischemia-Reperfusion Injury. This work was accomplished by Mingwei Tang, Yueying Mo, Zhishan Ling, and their colleagues from the Department of Rehabilitation Medicine, Zhujiang Hospital of Southern Medical. We will now introduce the current methods for identifying KLA sites, which are primarily divided into two approaches: liquid chromatography tandem mass spectrometry (LC-MSMS) and bioinformatics-based computational prediction. Experimentally, LC-MSMS serves as a core technique for detecting a wide range of post-translational modification sites. However, its application remains constrained by high sequencing costs and time-consuming procedures. With recent advancements in various predictive models, computational methods for KLA site prediction have advanced rapidly, becoming an important complement to MS-based strategies. Models proposed in recent years, such as FSAL-KLA, DeepKLA, AutoKLA, DeepClappered, EBFF-KLA, and PBIRD-KLA, utilize protein primary sequences as input and employ distinct neural network architectures to identify potential lactylated lysine residues. These models, having explored the methods for identifying KLA sites, we now proceed to the core of the article, the specific mechanisms by which non-histone lactylation influences cerebral ischemia-reperfusion injury. These mechanisms can be summarized into four key aspects: neuroinflammation, mediation of neuronal death, regulation of mitochondrial function, and mediating angiogenesis. In the context of neuroinflammation, non-histone lactylation exerts a dual regulatory role in cerebral IR injury by modulating the activation of glial cells, such as microglia, and the release of inflammatory factors. Regarding mediation of neuronal death, non-histone lactylation plays a dual role, either promoting or inhibiting neuronal death by regulating various programmed cell death pathways, including apoptosis and phoroptosis, during cerebral IR injury. In terms of regulation of mitochondrial function, non-histone lactylation dynamically and dualistically influences mitochondrial function and neuronal fate by regulating key processes such as mitophagy, mitochondrial dynamics, and oxidative stress in cerebral IR injury. Under the theme of mediating angiogenesis, non-histone KLA can alleviate. Currently, several proteins including p-SENBI, CBP, and ARS2 have been reported as writers, while others such as SIRT-SYN, HDAC, and KT-BAT have been identified as erasers of non-histone KLA. Based on the underlying mechanisms, we now focus on potential therapeutic strategies, which can be categorized into three aspects: regulation of lactate levels and transporters, regulation of non-histone lactylation modifying enzymes, and other specific regulatory mechanisms of non-histone KLA. While regulation of lactate levels and transporters can indirectly influence non-histone KLA, directly targeting non-histone lactylation modifying enzymes represents a more promising therapeutic approach to alleviate cerebral IR injury. The regulation of non-histone KLA involves versatile modifying enzymes such as writers and readers, and targeting readers which may offer higher specificity could provide a novel strategy. Crosstalk between KLA and multiple PTMs can be categorized into four distinct modes: collaborative modification, induced modification, progressive modification. This review innovatively summarizes current bioinformatics-based computational prediction for predicting KLA sites, systematically summarizes the temporal dynamics and diverse alterations of KLA following cerebral IR injury, and prospectively outlines development of potential therapeutic targets by focusing on non-histone KLA in cerebral IR injury. The effects of non-histone KLA are contingent upon the pathophysiological stage, varying with the duration of IR, the specific animal model used, and the timing of sample collection. This review summarizes bioinformatics-based computational prediction for predicting KLA modification sites by which non-histone KLA contributes to cerebral IR injury. Extensive crosstalk exists between KLA and other PTMs, which can be categorized into four mechanistic modes: collaborative modification, progressive modification, induced modification, competitive modification. These interacting modifications collectively influence disease progression. Targeting regulation of lactate levels and transporters, as well as non-histone lactylation modifying enzymes, represents a promising therapeutic strategy for cerebral IR injury. Current research on the role of KLA in cerebral IR injury remains limited. Further studies focusing on non-histone KLA hold promise for deepening our understanding of the underlying disease mechanisms